Hi everyone. Here I'm explaining about Q series guideline. Q series guideline in ICH is very important to understand the quality of the our products. If it is very helpful for if you belong to the pharmaceutical industries and if you're pursuing the pharma biotechnology and life science, it is very helpful for you if you're watching this video. Okay, let's discuss about what is Q series guideline and what is the importance of this. I'll explain in details. Let's it is quality guideline mainly divided into 10 subdivision. Q1 includes stability, Q2 analytical validations, and Q3 impurities, Q4 pharmacophobia, Q5 quality of the biotechnology products, or Q6 specification, Q7 good manufacturing practice, Q8 pharmaceutical development, Q9 quality risk management, Q10 pharmaceutical quality system. Let's discuss about the Q1. Q1 guideline also further subdivided into six Q1A, Q1F, Q1A. Stability testing of the new products, substances, and product provides evidence on how the quality of the drug, substances, or product varies with the time under the influence or environment factor. Q1B photo stability testing. New product, substances, or product evaluate the light sensitive. Some product has the very light sensitive. We need to check. We need to check that things. It comes under a Q1B or stability of the drug substances products. Q1C stability testing of the new doses form provide evidence of the quality stability of the new formulation of the approved medicines. Q1D bracketing or matrix design for the stability testing of the new drug substances or products. Bracketing it is designed to stability schedule such that sample are tested at all time on the extreme of the factor. The design assume that the stability of any intermediate level of the level is represented by the stability of the stream tested matrixing. It's the design of the stability schedule such that the selected subset of the total number of sample are tested at a specific time point for the all factor combinations. Q1 Q1E evolution of the stability data guideline addresses the evolution of the stability data that should be submitted in registration application for the new molecules identity, establishing shelf life for the drug substances product intended for storage at or below the room temperature. Q1B Q1F stability data package or registration application in climate zone. Third or third fourth describe the harmonized global stability testing requirement in order to facilitate the access access to medicine by reducing the number of different storage condition. Okay, let's discuss q q2 analytical validation. The objective of the this validation of the analytical procedure is to demonstrate that it is suitable for the intended purpose. It gives the validation parameter needed for the variety of analytical method and characteristics that must be considered during the validation. The type of analytical procedure to be validated: limit test, identification test, characterization include accuracy, precision, detection limit, quantitation limit, and detection limits. Okay, and Q3 impurity. Q3 guidelines are further subdivided into three: Q3A, Q3C. Q3A impurity in new drug substances. Q3B impurities in new drug products. Guideline addresses the chemistry safety aspect of impurity. Impurity are three types: organic, inorganics, or residual impurity. Chemistry aspect include a classification, identification of the impurities, listing of the impurities, generating reports, etc. And safety aspect include a specific guidance for qualifying those impurities that were not present or were at lower level in the batches of the drug substances or product used in safety or clinical trial. Q3C, impurity in the residual solvent prescribe the limits of the certain solvent. Q4, pharmacopoeia. Q4 describe a process for the evolution of a recommendation given by EWG for EWG for the selecting pharmacopoeial text to facilitate their recognition by the regulatory authority for the use in different ICH region. It includes mainly test microbial examination for non-sterile products and test for particulate examination, disintegration and dissolution test, uniformity test, sterility test, tablet friability, bulk density, tapped density of the powder, like that. Q5. Q5 of the biotechnology product. Q5A divided into Q5A, B, Q5E up to Q5A. It's concerned with the testing evolution of the viral safety biotechnology product derived from the cell line of the human animal origin, provides the general framework for that virus testing experiment. Q5B, it concerned with the analysis of expression construct in the cells used for productions of the rDNA derived product. Q5C is concerned with the stability testing of the biotechnological product. Q5D, it's concerned with the derivation or characteristics of cell substrate. Q5E is subjected to compatibility of the biological changes in their manufacturing process. Q6 specification. Specification is related to specify new product, new drug substances or products. Specifications are the key of documentation for the worldwide product license applications. Main object of this guideline is to be established set of specifications of drug substances or products. Okay, and let's discuss Q7. Q7 is, I think many people is aware about this. Q7 is the good manufacturing practice. This document is intended to provide guidance regarding the GMP for the manufacturing of API under the appropriate system for managing the quality. It also intended to help ensure that API meet requirement for quality and purity. Q7 mainly involve following API organizations personnel. Q7 mainly involves the following activities like API, active pharmaceutical ingredient, organization or personnel, building on facilities, process equipment, documentation records. Q8 pharmaceutical development. Q8 intended to provide guidance on the content of pharmaceutical development of the drug products. The aim of Q8 is to design a quality product and its manufacturing process to consistently deliver to intended performance of the product. And Q9, many people aware about this thing. Q9 basically is quality risk management. It offers systematic approach to quality risk management. This guidance provide a principle and tool for quality risk management that can be applied to all aspects of the pharmaceutical quality, include development, manufacturing, distribution, inspection, submission, on review process of use of the raw material, solvent, excipient, packaging, labeling materials used in the drug substances or drug products, basically. And final, Q10 pharmaceutical quality system. Pharmaceutical Q10 establishes a new ICH guideline describe a model for the effective quality management system for the pharmaceutical industry. Comprehensive model for effective pharmaceutical system is based on international standard organization ICO quality concept, which also include the GMP regulation. Okay,